Your career begins here at Satyabama Institute of Science and Technology, a deemed to be university, a nationally recognized and globally acclaimed destination to accomplish your academic aspirations. The Colonel Dr. JPR Research Park, along with the International Research Center, houses advanced research facilities that have 13 centers of excellence, 10 plus technology transfers, 100 plus patents, 10,000 plus reputed publications. The Satyabama Center for Advanced Studies is a state-of-the-art facility built with the aim to cater to the futuristic aspirations of our students with the aid of a metaverse studio, drone labs, maker's space, media center, advanced characterization facilities, ICT-enabled classrooms, and supercomputing laboratories. The engineering departments are equipped with sophisticated infrastructure, fulfilling the academic and hands-on requirements of our students. The architecture department is furnished with sophisticated design studios and spacious drawing halls, all of which support the learning process of our postmodern generation architects. The Satyabama General Hospital, along with the School of Dental, Nursing, Pharmacy, and Physiotherapy, have advanced clinical facilities. The monumental Central Library houses a diverse collection of books, periodicals, and journals. Additionally, it also has an open-air theatre with a seating capacity of 1,000. The Management, Arts, Science, Law, and Hotel Management departments comprise of all the necessary and required facilities, creating the perfect ambience for our far-sighted students and catering to their skill set needs with the end goal of equipping them to live their dream. Our chain of laboratories with practical exposure equips our students to build a successful career. Our institution has a team of dynamic and outstanding teachers who are ever reachable to our enthusiastic students, promoting the spaces to learn and places to grow. Greenery around the campus greets and accommodates our students, who are the backbone of a developed nation, edging them on to achieve the summits of their success. Satyabama Institute of Science and Technology.
Good afternoon, everyone. A warm welcome to all the participants to another session of Ocean Research Knowledge Series 2026, organized by the Center for Ocean Research and National Facility for Coastline Marine Research, Satyabama Institute of Science and Technology, with the support of Ministry of Earth Sciences, Government of India. Today's expert lecture is on titanium oxide nanoparticle toxicity in marine microalgae. We are very pleased to have with us Mr. Manikandan S., Technical Associate, National Facility for Coastline Marine Research, Satyabama Institute of Science and Technology, who will be delivering today's lecture. A very warm welcome to you, sir. Now I invite you to take over the session. Over to you, sir. Thank you.
Thank you, Sumesh. I'll proceed for this. Is my screen visible, Sumesh?
Yes, sir. Yes, sir.
Okay. A warm good afternoon to one and all present here. So the topic for today's presentation is titanium oxide nanoparticle toxicity in marine microalgae. So this entire topic will be covered from the basic cellular stress to photosynthetic dysfunction of microalgae in response to titanium oxide nanoparticle exposure. So I'll be presenting over the basic from the basic details like how the titanium oxide nanoparticle is getting interacted to what are the reactions or exposure reactions happening within the microalgae from cellular to photosynthetic responses. So before getting into what are the research works that has been carried out, we have to understand why we have to study the nanoparticles as well as the microalgae interaction, why it is essential to study those interactions happening within the environment as well as in the ecosystem, as well as within the cellular responses. So nanoparticles, like with the current age of industrialization, we cannot control the nanoparticle usage. Nanoparticles are tend to be produced in large quantities in different forms as well as in the different chemical states, and they have been increasing in number rather than getting decreased. They are tend to be increasing with the increased functionalization in various applications. Like the current focus of this particular study is titanium oxide nanoparticles. We have chosen titanium oxide nanoparticles as our target nanoparticles because they have been increasingly utilized in various industrial applications as well as in cosmetic applications as well as in homemade applications also, like in common sunscreen to paints. Titanium oxide nanoparticles has been found to be increased quantity, and with this increased usage there has been considerable amount of environmental release also over the period of time. The particles that are associated with these applications tend to release those particles into the surrounding environment, that may be land, soil, as well as the water or in the aquatic ecosystem also. These released particles will cause an adverse effect as well as some beneficial effects to the surrounding environment based on the quantity as well as depending on the concentrations of the released particles in the surrounding ecosystems. So to understand the nanoparticle microalgae interaction, we need to identify the widespread use of nanoparticles as well as the associated environmental release. And the reason we have chosen the microalgae as a target organism is they are the primary producers. Without microalgae in the ocean as well as in the freshwater ecosystem, we cannot imagine how the world gets sustained because they are the primary producers. They have to fix the carbon for the upcoming other organisms or other higher level organisms. They are the only primary producers. They have to fix the carbon and have to be the energy source for the other level of food chains. And there has been potential ecological risk if the released nanoparticles are affecting the primary producers. There may be a shift in the shift in imbalance of the ecological food chains also. So we have to identify the associated ecological risk within this microalgae, and there are knowledge gaps like how this nanoparticle, we in general people to know that how the nanoparticles are getting affecting the primary producers, but most people won't understand like how these nanoparticles are getting modifying the metabolic pathway or modifying the morphological responses that are happening within the microalgae. So those studies also has to be carried out. And like I said in the beginning, like the reason for selecting microalgae is like any ecological indicators or ecological pollutants that are getting released into the environment. The first point of contact for the targeted pollutant will be the microalgae, because what are the damages that are happening with the microalgae will get magnified and transformed into the other state of food chains. So they form the base level for the corresponding or upcoming biotransformation application. So what are the adverse effects happening within this microalgae has to be studied, understood, and has to be implied to the other food chain levels. So like I said, microalgae are primary producers and they play a major role in carbon sequestration, and they form the base of the marine food webs. Like how these particles are getting transformed from one to other side, and they also have a role in nutrient cycling. Apart from this environmental role, they have also have industrial applications, like they play a major role in biotechnology industries, mostly in downstream processing industries, as they have value added products like lipids, carbohydrate, proteins, and they can be used for applications like biodiesel production, pigment production, fatty acid productions also. So what are changes that are happening with this doesn't stop at this level. They either get transformed or biomagnified to the other levels. And the other reason for selecting titanium oxide nanoparticles is they are produced in enormous amount due to their photocatalytic activity or its advantages being utilized in industrial applications. Titanium oxide nanoparticles in general exist as two forms. One is anatase form and another one is rutile forms. Compared to rutile form, anatase feels more stable and they have more photocatalytic activity. So that's why anatase form of titanium oxide nanoparticles have been utilized in many industrial applications, starting with sunscreen, cosmetics, plastic coatings, and other many applications. And the route for the environmental entries, the produced products with the nanoparticles, once their use or their lifetime has been expired, they will be dumped into the either in the landfills or transformed into the ocean or in the aquatic ecosystem. From this, due to the environmental degradation of sunlight, harsh conditions of the ocean, the particles that are incorporated within those products get leached out into the environment, and there is a persistent risk associated with the titanium oxide nanoparticles. And why it matters is because the increasing presence of the titanium oxide nanoparticle utilization poses a risk that this particular nanoparticles are continuously released into the aquatic ecosystems. And before getting into the detailed aspect of what are the things that are carried out, we have to understand like how these nanoparticles have can attack those microalgal cells. Keep out the titanium oxide nanoparticles or isochrysis galbana aside. Like in general, nanoparticles, they are like a chemical pollutant. Nanoparticles are one kind of chemical pollutant, and microalgae as a living cell. Once this nanoparticle gets interacted with the microalgae, there are like 4 to 5 mechanism, like what are the things that can happen, starting with the uptake. Like cells has to adhere, this nanoparticle has to adhere to the cell surface. Once adherence has been completed, they have to be uptaken by the microalgal cell. Once the cells nanoparticles are getting allowed inside the microalgal cell, there will be cellular responses varying from photosynthetic responses, oxidative stress responses, and metabolic effects. And in adverse, they can also cause genetical effects also. These are the 5 different mechanisms, like how the particular nanoparticles and particular algal cells interact and what are the effects are happening, starting with the absorption, cellular uptake, oxidative stress induction, followed by photosynthetic interference. And in adverse effects, there can also be genotoxicity and metabolic effects. So the research question and hypothesis that we can infer out of this particular study is like how this nanoparticles inhibit the growth. We can say that nanoparticles affect the microalgae, but what are the things happening within the microalgae? How does the growth get deteriorated? And what are the oxidative states? How does the uh, proposed nanoparticle induce the oxidative stress that are happening within those microalgae, and how does the nanoparticles influence the metabolic profile or photosynthetic parameters that are happen, whether these nanoparticles are following the time dependent or concentration dependent pattern, and in whole what is the underlying mechanism that is happening between the nanoparticle as well as in the microalgae. These are the research questions that we have answered, and hypothesis that we have hypothesis that acute toxicity are the most adverse form of toxicity that is happening with the pollutant as well as the microalgae, and all these are happening through the oxidative stress, like they are the underlying mechanism to which all the toxicity effects are happening. And this is how we have framed our methodology. We started with the identifying our target pollutant as well as the target algae. In this case, we have taken titanium oxide nanoparticle due to their extensive application, as well as we have taken Isochrysis galbana as our target microalgae. This is because Isochrysis galbana is one of the common microalgae that is found in all aquatic ecosystem as well as in the marine ecosystem. And rather than having the single environmental role, they also have a industrially important application, as they are lipid rich microalgae, and they are also known as brown algae also. So this is the reason that we have taken this titanium oxide nanoparticle as well as my Isochrysis galbana as our target organism. This Isochrysis galbana we have isolated from JPR Fishing Harbor, Muttam, through extensive sampling and plating procedures. We have collected the water samples. From the water samples, serial dilution, subsequent plating processes have been done to isolate the pure culture of Isochrysis galbana and maintained in our Ocean Research Lab, Center for Ocean Research Laboratory. And in case of titanium oxide nanoparticle, we have procured the commercially available titanium oxide nanoparticle in anatase form. This is because we have to identify what is the real implication that the commercially available nanoparticles having on the our microalgae. And once this microalgae and nanoparticles are ready, we have fixed the concentration based on the environmental relevant concentration. Based on the previous study, we have identified what are the concentration that have been reported in the previous studies in the environmental ecosystem. Based on that, we have fixed the concentration from 10 mg per liter to 50 mg per liter. This 50 mg per liter represents the maximum futuristic concentration that is supposed to be existing in the aquatic ecosystem. So once this concentration has been finalized, we have carried out acute ecotoxicity studies based on the OECD guidelines. We have taken 5 different concentrations at 4 different time intervals ranging from 24, 48, 72 and 96 hours. This represents the acute endotoxicity studies. These studies we have represented in, like, these are the parameters that we have estimated through this studies, starting with growth inhibition, like what are the growth inhibition that is carrying out within this because of this toxicity exposure. Once this growth inhibition is done, we proceed to estimate the biomass quantification, like we can identify, like we can say growth has been inhibited, but what is happening intracellularly. So we determine the biomass as well as the biochemical composition, carbohydrate, protein, lipid, and we have calculated the photosynthetic pigment, chlorophyll, carotenoid, as well as the photosystem parameters, PS2 photosystem 2 parameters, membrane integrity, lipid peroxidation, oxidative stress, morphological changes, as well as the statistical analysis. This is the cumulative analysis that we have performed in this. In going slides, we will explain what are the parameters in detail and how it is affecting the microalgae. So we started with the characterization. Even though we have procured it commercially, we have to estimate, confirm that these are the chemical state and what are the particle sizes of the particular nanoparticles. So we started with the particle size analysis using dynamic light scattering application, as well as the SEM, XRD and SAED, selective area electron diffraction analysis also. All these are the physical characterization of the particular nanoparticle, and this confirms that nanoparticle exists in the nano size range of 54.8 nanometer. That is the maximum, like average diameter of the existing nanoparticles, and the nanoparticles are in the heterogeneous form, like they don't have the uniform shape or size. They are existing spherical form, but the size and shape differs from each particle to other particles. And the XRD also confirms the chemical state, like these chemical patterns represent the particular chemical state of the nanoparticle, especially in the annotated form, and the same has been reflected in the SAED patterns also. The patterns what we have obtained in the SAED patterns as well as the XRD confirms the physical nature as well as the annotated form of the particular nanoparticles. Before getting into those analysis of growth inhibition, we have to identify whether the particular nanoparticle as well as the microalgae, when introduced together, they getting interacted or whether they are getting settled down. So what we have did is we have planned 4 experiments, 4 groups. The first group will contain only the algae. Second group will have algae, only the titanium oxide nanoparticles. Third group will have algae and nanoparticles together, and fourth group will have algae and nanoparticles separated. We have calculated the OD values at frequent intervals and plotted as a settling curve. This curve indicates that whether the nanoparticles and algae are getting interacted at efficient intervals or they are getting settled separately. So the green line indicates algae, and the corresponding brown line, red line indicates the corresponding experimental groups, and this says that algae in the separate groups don't have that much settling. They are always available in the suspension forms, but titanium oxide nanoparticles in the individual groups they tend to settle. They don't form the dispersion. They form the colloid or suspension solution, and they get settled at the bottom. So algae, titanium oxide nanoparticle has the fastest settling rate, but when algae and titanium oxide nanoparticles are introduced together, the settling rate of the nanoparticles comparatively becomes lesser than the individual nanoparticle group. This is because algae nanoparticles has the EPS protection group, and these biochemical compositions tend to trap the nanoparticle within this biochemical matrix, and the settling rate tends to slower. So this experiment part confirms that nanoparticles are getting well interacted within the microalgae. So the toxic effects that are happening within the microalgae are due to the nanoparticle itself, not with the surrounding environments. So we started with the growth inhibition studies. So like once the nanoparticles are getting interacted with the microalgae, we collected the cells at regular intervals 24, 48, 72 and 96 hours, and we calculated the cell growth. We counted the cells at the regular interval and presented at cell count as well as the growth inhibition percentage as well as the dose response curve and the specific growth rate. We didn't stop at the calculating, simply saying that cells growth has been diminished. We didn't propose that. We calculated what is the cell count, actual cell count, and based on the cell count, we calculate what is the percentage inhibition that is happening within those microalgae, and whatever the specific growth rate difference between the time intervals, like 24 hours to 78 hours, what is the specific growth rate. So all this says that this particular microalgae forms the time dependent and growth concentration dependent. So with the increasing time and within the increasing concentration, algae growth tends to be substantially decreased as they possess toxicity to the microalgae. You can see from this cell count data, like the cell growth at the control, like without any microalgae, they tend to reach up to 2.5 into 10 power 7 cells per ml. But when interacted at the highest concentration as at the higher time interval, uh, the cell growth has been subsequently decreased. That has been reflected in the growth inhibition as well as in the specific growth rate also. And in coming to the biochemical composition, we started estimating the biomass dry weight. Like we collect the samples at regular time interval, and we calculate the dry weight through gravimetric filtration method, and we calculate the dry weight. So the control cells without any treatment or any exposure to nanoparticle will have the pristine growth, and the biometric biomass will tend to increase at the regular time intervals. But the other experimental groups, when interacted with the algae, based on the concentration as well as the time, there is a subsequent decrease of the biomass growth, dry weight concentration also. So this particular parameters also reflects the data that we have obtained to the cell count. So without the cell propagation, we cannot say that this biomass will also be more in amount. So cell count has also diminished, so that biomass has also been decreased. We didn't stop at that level. We proceeded to the estimation of carbohydrate, protein, and lipid concentration. So what are the things that are happening intracellularly? We have estimated Carbohydrate we have performed it through phenyl sulfuric acid method. Protein we performed to bovine serum albumin method, Bradford method, and lipid we estimated gravimetrically through Blick dryer method. So what are the biomass that have been obtained? We added the phenyl sulfuric reagents and calculated the concentration of the particular carbohydrate through and estimated it through the standard curve of glucose. Same for the Bradford method, we estimated through the standard curve of bovine serum albumin. And for the lipid concentration, we estimated it gravimetrically through Blick dryer method. So all these data corroborate what are the findings we have obtained from the biomass as well as in the cell count. With the diminishing biomass as well as the cell count, the carbohydrate, protein, lipid concentration tend to decrease with the dose dependent and time dependent manner. And photosynthetic pigment, this is where we have observed a significant result. Like we have estimated two photosynthetic pigment. One is chlorophyll and another is carotenoid pigment. These two photosynthetic pigments play a major role in photosynthesis. Estimating them and quantifying them will have an implication in the photosynthetic mechanism that is happening within the microalgae. So we estimated through ice cold ethanol methods. So when seeing with the chlorophyll concentration tend to decrease with the increase in the concentration of the nanoparticle as well as in the time. But when it comes to the carotenoid, we can see the totally different approach. Like carotenoid concentration tend to increase with the increase in time period as well as in the increase in the concentration, because they act as the first level of stress mechanism. Like what are the stress mechanism that is happening within the microalgae, that will be reflected in the carotenoid concentration. So whenever a nanoparticle or any foreign particles that enter into the microalgae, there will be a stress caused by the oxidative stress responses. So those responses will be quenched by those photosynthetic pigments. These pigments are also called as the oxidative stress pigments, quenching pigments that will quench the stress that are caused by those oxidative stress responses. So the carotenoid concentration will be produced in more in response to the stress that is happening within the microalgae, but that will be happen only to the particular level, or beyond that the carotenoids will not be produced more because they have reached the threshold level. So this states that before getting into the complete cellular responses, photosynthetic pigments act as a primary defense level and try to quench what are the things that are happening within those microalgae. And not stopping with the chlorophyll and carotenoid, we targeted the photosystem too, because microalgae are photosynthetic organisms. So photosynthesis, the major role that is happening within the cells. So we have estimated the what are the things or impairment that is happening within the photosystem too. So like in case general photosynthesis of microalgae take place in the chloroplast, and there will be two systems, photosystem 2 and photosystem 1. So from photosystem 2 there will be a light dependent reaction starting with the conversion of water to electron plus OH minus ions, and that will be traveled to the photosystem 1 through subsequent electron carriers. From that photosystem 1 we will be taking in the ATP production and other production. So what are the impairment that is happening within the photosystem 2 will have a greater effect in the further metabolic process also. So we have given utmost care to what are the things happening within the photosystem 2. So we have selected four parameters. One is maximum quantum yield, Fe by Fm, photoinhibitory damage, Fe by F naught, electron transport rate, ET naught by TR naught, and absorption flux, ABS by RC. So these four parameters play a major role in those photosynthetic electron transport. Like whenever a water molecule is getting converted into electron plus OH minus ion, they have to be actively transported from the photosystem 2 to photosystem 1 through series of electron transporter like flastoquinone, cytochrome B6 and other reactors. So what are the damage that has happened here? They will not be able to transport efficiently. That we have determined through these four parameters. So maximum quantum yield is like how well this photosystem 2 is functioning and whether there is photoinhibitory damage associated and electron transport is efficient, or whatever the electron that is has been absorbed or transmitted to the further photosystem 1. All this indicates that there is a imparable damage within the photosystem 2, like the yield has been diminished, photoinhibited damage has also been increased, as well as the electron transport rate has seemed to be decreased. All these four has been indicated within this photosystem parameters, and we have proceeded to estimate like what are the oxidative stress parameters that is happening within the analysis. We started with the analysis of like how well the membrane permeability, whether there has been membrane damage, and what are the concentrations damage happening with the membrane, as well as lacto dehydrogenase and the ROS productions. So to summarize this, we can have a interpretation like whenever an pollutant leave out like nanoparticle or titanium oxide, whenever a harmful pollutant is getting interacted with the microalgae, the first layer of contact will be the cell membrane. So cell membrane has to be permeated, and the permeated particles has to enter into the microalgae, and they have to has the response. So we estimated like what are the level of damage that has happened with the membrane, and that membrane damage can also be corroborated with the findings from MDA. MDA is nothing but malondialdehyde assay. So they represent the lipid peroxidation that is happening within those cell membrane. So cell membranes are made up of phospholipid bilayers. So if the lipid membrane has been damaged, they can also be estimated to MDA. And once this membrane has been permeated or they have been compromised, there will be leakage of intracellular components. So one such component is lactate dehydrogenase. Lactate dehydrogenase is a intracellular component that is present within the mitochondrial gene. So if the mitochondrial organelle, if there is a damage to the cell membrane, those particles will also be released into the cytoplasm, and they will be also released in the surrounding environment. And ROS, in general, we didn't calculate about what are the superoxide or reactive other oxygen species. We in general we calculated the reactive oxygen species. So membrane permeability we estimated through cytox green assay. It's a fluorescence dye. If there are any deviation or any impairment within the membrane, they get penetrated into the cells and they will produce fluorescence. If the fluorescence intensity is higher, we can conclude that membrane has been permeated. Based on the fluorescence intensity, we have plotted the graph, and it also formed the same trend. And the membrane MDA concentration also follows the same trend, like lipid peroxidation is happening based on the time as well as the dose dependent manner, and same the LDH quantity has been increased in number as they have been released more into the surrounding environment, and the ROS production is also higher based on the time dependent as well as in the dose dependent manner. This is the overall mechanism, like how we have identified what are the things happening. Like said, there will be membrane damage. Once the membrane damage is there, the nanoparticles will be getting penetrated into the intercellular, and once they enter into the phase, they will target the photosynthetic parameters through chlorophyll's damage, and there will be subsequent damage through the mitochondrial too also. And in adverse effect, there can be damage to the there will be damage to genotoxicity effects or in the toxicity effects in the higher levels. So this is where we stand out from the other studies. We have calculated, estimated, and identified the highest level of damage that is happening within the photosystem 2. Like I said, the photosystem mechanism will have two spaces. One is photosystem 2 as well as in the photosystem 1. All this happens within the chloroplast pigments. So starting with the nanoparticle exposure, they will start their interaction in the water splitting chain. Like water has been splitted into electron plus OH minus ion. If there is a damage, the subsequent electron will not reach the photosystem 1, and they will have a greater damage in the ATP productions also. So this will scale up to the further metabolic cycle like Calvin-Benson cycle and other pathways. So starting with a similar very small damage of water splitting chain, that will have a magnified effect in the further chains also. And we have identified the morphological deformations what are happening within the microalgae based on the concentration. In control, you can see the cells are still in the intact pristine form. There is no much no too smaller damage to the microalgae, but within the increasing concentration, the deformations and the impairment is very higher. So 10 mg has a very few morphological deformation, but when you compare to the 50 mg per liter, the complete cells has been deteriorated and all the intracellular particles has been released. And all this data, whatever the parameters that we have identified and estimated, we corroborated it with the statistical analysis. We have correlated, like what are the parameters are positively correlated and what are the parameters are negatively correlated. Almost all the parameters that we have estimated as a positive correlation, except the carotenoid pigments, as well as in the lipid peroxidation. So cell count and other parameters, if they are diminished or negatively influenced, there will be a negative influence pattern on the carbohydrate, protein, lipid, as well as in the other parameters also. But if the cell count is decreased and they are causing due to the toxic responses, there will be increased concentration of oxidative responses. So there will be more amount of RS. So they are negatively correlated. That we have identified through the Pearson correlation matrix. And we have also performed the principal component analysis to find out what are the parameters that are closely aligned. So all this data we have performed to have the reproducibility. If and all in future some persons want to replicate the data or have to use the same methodology for their subsequent analysis, this data can be reproduced and can be scaled to their based on their experimental analysis. So to conclude, in general, we have correlated again, like not with the Pearson correlation, we have correlated with the so this whole study we have concluded that that's a time dependent pattern. With the increasing in time and as well as in the concentration, there has been increasing damage. To conclude that, we have calculated the strength of association, R square value for almost all parameters. So R square value for all parameters are in the range of 0.9. So this confirms that the concentration follows the trend and the damage also follows the trend and correlated with the ROS. So we have taken ROS as a basic parameter because they are the underlying mechanisms. With the ROS production, everything gets damaged. So we have correlated. So cell growth has been negatively correlated with the ROS. Biomass also negatively correlated. Almost everything are negatively correlated because if cell growth is decreased, there will be an increase in production of ROS. They will also have a subsequent damages in the ROS productions as well as in the cell growth. So to have an overall idea to why did we have carried out this study, what are the implications in the ecological and environmental aspects. So microalgae, as said, they are the primary producers. What are happening within those microalgae will get biomagnified and transformed. That has been indicated in the microalgal population and ecosystem. Microalgae will be consumed by the secondary consumers as well as the tertiary consumers. So if they consume the pollutant, if the secondary consumers consume the consume the microalgae that are interacted with the titanium oxide nanoparticle, they also get transformed. That will be transformed to the subsequent higher levels. So the ecosystem level consequences will be there. And freight and transport. So microalgae, if they have been uptaken by the microalgae, once they have been dead, they will get settled at the sediment. So from there will be uptake and transport of those titanium oxide nanoparticles to the subsequent organisms also. And there is a long-term risk associated with this microalgae. And in terms of environmental management, there has to be a need for the monitoring as well as the controlling. We cannot control the use of the nanoparticles, but we have to control the release of those particular nanoparticles into the environment and to assess the sublethal and the persistence of those nanoparticles in the aquatic ecosystem and to design, if possible, safe and green titanium oxide nanoparticle for the applications. And the take home message will be the growth has been inhibited and that has been reflected in the biochemical composition, that these data has been implicated in the photosynthetic mechanism as well as in the ROS damages. And we have said, like in the previous slide, there are more ecological and environmental implications also. To conclude this study we have carried out like 2 to 3 years back. That time only the nanoparticles as possessing a severe threat to the ecosystem. But with this growing conditions and in current conditions, not only the nanoparticles have surpassed the levels, and we have started estimating the levels of microplastic, nanoplastic, pH, temperature. So there are many anthropogenic pollutants that are causing damage to the ecosystem. So those parameters has to be assessed, and we have performed only the acute toxicity studies within the 96 hours. We haven't performed the chronic toxicity studies that we also take care in the future aspects. So the take home message that I will give to the participants is like microalgae, they essential for this sustaining ecosystem. So we have to utilize the nanoparticles in the sustainable way, and then and there we have to estimate what are the pollutants that are in the present in the sustaining ecosystems also. Thank you.
Thank you, Mr. Manigandan, for your deep and insightful talk on topic titanium oxide nanoparticle toxicity in marine microalgae. Participants, if you have any questions regarding the topic, you can post it in the chat box. Hope so there is no questions in the chat box. We can conclude the session today. So thank you all for your enthusiastic participation and for making ORKS 2026 a wonderful experience. As today marks the final day of ORKS, we hope you found the sessions insightful and the interactions enriching. And we are also happy to share that admissions are open for MSc Marine Biology program offering hands-on training, field exposure, and exciting research opportunities. Stay connected with our websites and social media pages for updates on upcoming programs, workshops, conference, and events. And thank you all.